now we have a very short talk. This is a lightning talk, right? From Kewalin Samart from USA. Are you there?
Yes, I'm here.
Okay. Hello.
Hello.
Can you share your screen?
Okay. Can everyone see my screen?
Yes.
Okay. Everyone hearing me okay? Everything's fine?
Yes,
okay.
I can hear you. Yeah.
Okay, so let me get started. Hello, everyone. My name is Kewalin Samart. I am an undergraduate student majoring in computational mathematics with a minor in computational math science and engineering at Michigan State University. Today, I'm really pleased to present our recent work on reconciling multiple connectivity score for drug repurposing. I work on this project with Phoebe Tayashime, Dr. Stephanie Levsicki, Dr. Ajun Krishnan, and Dr. Janani Ravi. To begin, I would like to talk about computational drug repurposing. So computational drug repurposing is an efficient alternative for drug discovery since it is both cost and time effective compared to the traditional ways of discovering new drugs that require a significant amount of time and budget. So instead of doing that, computational drug repurposing helps us identify new drug indications based on the existing drug that we currently have. So as the Derivate Lab, we focuses on developing computational approaches to study infectious diseases. And one of our ongoing project is drug repurposing in infectious diseases. And our project basically is gene expression-based drug repurposing. So we compare similarity between disease and drug gene expression profiles to get their similarity level and use the similarity level to get the ranked drug list, which basically a list of efficacious drugs that we predict as a drug that can successfully reverse the disease signature. And the key concept of connectivity mapping is that we compare disease and drug gene expression profiles started with a ranked order disease gene list, where we have a list of genes with disease perturbation data, and then we rank all the genes based on their gene expression value in descending order. Then we separate the genes in the list into up and down regulated gene set. And then we use this disease gene set to compare similarity with the drug gene list. And then after that, we will quantify relationship between disease and the drug. And the cases when we have reversal relationship, basically reveal like the relationship that the drug can successfully reverse the disease is the two cases in the middle here. When we have upregulated disease genes distributed at the bottom of the drug profiles and the downregulated disease gene distributed at the top of the drug profiles. And last year, when my teammate and I were reading paper after paper and trying to figure out which method for computational drug repurposing using connectivity based, which one is the best approach that we should use in our project. And we have found that many methodologies in the past 15 years have been developed toward finding reversal relationship between disease and the drug. And this is a timeline indicating all of the connectivity score that have been proposed by different studies within the past 15 years. And then we have found some issue with the proposed studies in the past 15 years that they have been described all the method using inconsistent notation and terminologies, although they have some kind of like shared set of mathematical and statistical concept and idea, which make it very hard for us to interpret and understand the connections among the different score. Also make it very difficult for us to compare, benchmark, and build on the existing methods. Therefore, we started thinking that if we reconcile all of the score using a new unified scheme by reconcile everything using consistent notation and terminology, it would be easier for us to both understand and interpret and also compare, benchmark, and build on the existing one. So that's come to, okay, so this is some of the example, the case that we have inconsistent notation and terminology described in different paper. So for example, in GSCA paper, they refer to the enrichment score as ES, but in CMAP 1.0 paper, they refer to the same enrichment score, but use KS instead. Another example would be number of drug genes in the drug gene list. In GSCA paper, they describe it using capital N, but in CMAP 1.0, they describe it using a lowercase n. So we think that it would be very helpful for us to redefine all of the score using our own notation. And this is a general notation table in our paper where we list all of the common notations to use across all different methods. And this is some of my notes from last year when I was trying to figure out what are the relationships between all of these different methods that have been proposed within the past 15 years. And as you can see, that is kind of a myth when we try to establish the connections among them using mathematical and statistical insights. And we redefine all of the method using our own notation with like, but for today, I will walk you through how we can relate the popular GSCA method and CMAP 1.0 method in insight into like the mathematical details as an example. So for the gene set enrichment analysis GSDA, we found that the similarity matrix that they compute, basically they compute it using weight size to sample Kolmogorov-Smirnov statistic. And CMAP 1.0 method, basically they based on the GSCA, but it's kind of like the adapted version of the GSCA. Instead of using the two sample Kolmogorov-Smirnov statistic, they use unweighted size one sample Kolmogorov-Smirnov statistic. And we have found that and realized like this effort can be very helpful, not just for us that doing a computational drug repurposing project, but it can be very helpful for the community of computational drug repurposing researchers if we like redefine all of this and publish this work. So basically, this is a taxonomy of connectivity score that we finally constructed as a result of our work. So we have established a layout every disease drug connectivity score, disease drug similarity matrix, and connect them based on the relationship in the terms of math and statistic concept. So as you can see here, the first column is disease drug similarity matrix. The second one is disease drug connectivity score. The difference between these two is that we will use disease drug connectivity score to quantify the final result for our predicted drugs. But the disease drug similarity matrix is kind of like the basic concept of computation where we start to quantify relationship between disease and drug. And then we use the disease drug connectivity score that computed based on the similarity matrix as the result to quantify the drug candidates. And in panel B, it does some formulation with that we reconcile using our own notation. And for convenience to the reader, we also provide notations table like this for the reader if they want to go back to the previous study based on our current notation that we define in the paper. Basically, we have this notation table along with every single method that we mentioned in our work. And we have found that different score can be easily interpreted by looking at the size of similarity matrix. Basically, the first three rows here are the cases where we have favorable outcome, where the score gives us like the reversal relationship between disease and the drug. Basically, these three cases, the reversal relationship, we can observe it by looking at the negative enrichment score for disease upregulated genes or positive enrichment score for downregulated genes, or we can looking at the negative score of the pairwise similarity measures. And these three scenarios would correspond to the two cases where we discussed earlier in the talk. And not only the relationship or connection that we establish using our effort, but we also get some biological and practical insight of the score as well. The first insight that we will talk about today is nature of reversal. The second one is the choice of threshold for disease, for input disease data. The third one is the amount of input information. And then we will talk about the symmetry between disease and drug data. For the nature of reversal, we have found that the stronger connection, the stronger reversal relationship between disease and drug can be determined by the lower negative score of the connectivity score. And for the choice of the threshold for input disease data, where we separate the disease list into up and down regulated genes, we can do this by using p-value cutoff or fold chain cutoff, or rank all the genes based on their different genes expression value. And all of these choices, which one that we use will finally affect the performance of the connectivity score. The next one is the amount of input information and symmetry. We have noticed that each of the similarity metrics, they require different amount of disease information and drug information as an input. And this difference in input also affect the symmetric property of each of the score. So basically we have found that the score with symmetric property would, we can use the score with symmetric property to quantify not just disease and drug relationship, but also disease relationship or drug relationship as well. And according to the previous studies that we summarized, exam and XCOS perform better than the enrichment score-based method. The eMudra score as a combination of multiple metrics outperform any single metrics. And the performance of the enrichment score and CSS depends on the number of genes in the disease signatures. And the rank-based method usually can suffer from the distribution genes with a high rank, but not significantly differentially expressed. And in conclusion, the method proposed in the past 15 years have been built upon each other over time, thus addressing previous limitations, leveraging larger amount of data, achieving better performance in prioritizing repurposed drug candidates for diseases. And they also have shared number of core conceptual and analytical ideas, also use similar statistical techniques and quantities. However, these different studies describe the method using inconsistent notation and terminologies when naming system refer to previous methods and mathematical details, which is an impediment to cogent detailed understanding of current method, their transparent benchmarking and evaluation, also the development of newer methods that continue to build on existing one. With our unified scheme, we can improve the consistency of the future method with the existing ones, and also can help clearly establish the origin of the analytical idea, which make it easier for us to compare, benchmark, and build on in the future. So for the continuous and transparent incorporation of the newer method, apart from our traditional paper, we also host GitHub repository and coupled with a live document for any researcher in the community to build on and put changes to. And lastly, I would like to acknowledge everyone in the J. Ravi and the Krishnan labs at Michigan State University for their support and feedback on the work, especially Dr. Janani Ravi and Dr. Ajun Krishnan, both of my fantastic mentors, for their guidance and support. Also, I would like to thank all the funding and the conference for the opportunity to present my work today. Thank you so much.
Thanks a lot. We have time for questions. I have one question.
Okay.
When you define the strong and weak connections, and you define the strong as negative and the weak as positive with blue and red color, it's not clear for me how does it relate with the disease and drug score that you have, and it's G positive, G negative. For me, it was contradictory. Maybe you can explain this.
Okay. Which slide? This slide?
Go back. There. No.
This one?
No, next.
Okay.
No.
Okay. I think it must be this slide, right?
Yes, but the
reversal relationship
where you have the strong and weak, the strong and weak and weak.
This slide, right?
Yes.
Okay.
Yes.
Okay. So what I was saying is that the score that revealed the stronger reversal relationship would be the score with lower negative. Yeah.
Okay, but and the next map. Okay, so that's why I was confused, because you have this positive as blue and negative as red here, and in the other side you have negative as blue and here as red, and then I was just confused.
Okay.
How does it relate, you know?
So basically the nature of reversal, like the scale that I'm showing here, is just like the final result that we interpret like the relationship between disease and the drug. And this is like the start point where we compare disease and drug genes. So basically it's just like a process that we need to do is that we rank all the genes based on the expression value first, and then separate the genes based on the size of the expression value that assigned to each gene. Okay. And then we use this set to compare and get the similarity between disease and drugs. And then this would be the final result, like the final score that we get. And the lower negative score of the final score would interpret the stronger reversal relationship. Yeah, so basically this positive and negative side here, just the side of expression value that assigned to each gene.
Okay, yeah. Now I got it.
Okay.
Okay. Thanks a lot. There is another question. Carlos Lagos say, one of the main obstacles for drug repurposing are the drug ADME tox properties linked to the pharmaceutical form used, for example, eye drops or are injectable. Do you think that the identification rate for drug repositioning could be improved by including pharmacokinetic parameters or connectivity descriptor typically used to measure drugs' physicochemical properties?
Yes, and that actually is our future direction for our project, where we think that we are going to look into more insight about the genes and also like pharmaceutical details later. But for now, we are integrating all of this method and try to see which drug coming up the most, and then we will look into the details like in biology later after this. So it's kind of like the future direction that we are looking into. Yeah.
Okay, thank you. The last question is from Juliana Murillo. Juliana, can you open the microphone?
Thanks, Wendy. Well, first of all, thanks for your presentation. It was really clear, well ordered. Also, it was very interesting the way that you present the timeline and all the different transformation that you perform in order to unify and present like a unique way for name all the different scores and all the different metrics that all their methods use in this context. I just have two short questions. The first is related with the amount of the different notations that you are presenting in your new method, because I saw, I know, in one on your slides you show like the older notation and the newer notation, and I saw that there is almost the same number of notation, but just presented in the different way. I don't know. My question is, if you really think that that you are presenting a fewer notation or a newer way in order to have like a compilation or a reduced amount of notation that the newer methods can use in order to have a common language when you present those new methods. And the other question is, what do you think that will be like the recipe that the new method should follow in order to keep the newer notation that you are presenting in your work? Will be, I know, the hints or yes, the notation that the newer method should follow in order that all of us have a common language when we talk about those methodologies.
Okay, so I would like to answer the second question first. So the common language that I think it would be the core for connectivity score is that for the connectivity-based drug repurposing, basically we started with disease and drug information. So I think the common language that we need to follow in order to get the unified scheme in the future is the notation for disease genes and drug genes, and like number of disease in the drug profiles, number of genes in disease profiles, and also let me see, the general notation would be more helpful. Yeah, and it's like the way that we have the input information for disease and drug, like the way that we rank whether that method needs the list of drug or just the regular set of drug genes. So as you can see here, like we have some notation with the arrow above. So basically this indicates that the input needs to be ranked based on the expression value of each gene. And like some notation, just like the regular one without this, basically indicate that we can use that a set of gene, not a list that rank based on the gene expression values. And I think that would be the things that we need to account for the common language for the newer approach. Yeah.
Thanks, Kewalin.
And could you repeat the first question again? I'm sorry.
The first question is if you really think that you are presenting a lower amount of new notations in order to get that unified code, or if maybe you are just renaming the older scores that presents the older methodologies. Know if I clear? No?
Are you talking
for example,
about...
if you go for example, in that slide, I know I also saw more newer notation than the older ones, and I just thinking about if that new method is really, if you think that it will be really clearer for the for the other people that are developing newer methodologies, or maybe if the more important results behind your work is just compiling and comparing the older methods and proposing a combined way to to name the different scores.
Sorry, Kewalin, can you answer Juliana by chat to finish the talk, because
Yes.
we have a short time for the next talks that will start at 4:30, so in 10 minutes, and I would like to invite the people for a break, and then you can turn off the cameras and microphone and go to the chat.
The chat. Okay.
Possible?
Thanks,
All
Wendy.
right. Thank you.
Okay. Thanks a lot.